I am going to discuss ELISA. As it was mentioned in my previous video of the D dimer. There I use the term ELISA. ELISA. It is a very important topic for all the students who are directly or indirectly involved with the pathology in which either you are pursuing, performing or studying the immunoassay. Any kind of test which are required to be done quantitative method, ELISA will be necessity of that procedure. Here we will start discussing ELISA. What is ELISA? ELISA stands for enzyme. Wait a second. Enzyme-linked immunosorbent assay. Okay, enzyme-linked immunosorbent assay. It is a biochemical technique used to detect and quantify the proteins, hormones, antibodies and antigens. So here it can be said as it is a biochemical technique. For what? To quantify and detect what? Hormones, antigens, antibodies, and proteins and processes. It is biochemical technology used to quantify and detect protein, hormone, antigen, antibody. Okay. It is based on antigen-antibody interaction with an enzyme-linked reaction producing a measurable signal. It means here antigen and antibody bind to each other and produces basically a signal, and this machine or ELISA reader find out and calculate those signals and convert them in digital values. Let's discuss a little history of this ELISA. Okay. The ELISA, before ELISA, there was another method which is known as RIA, radio immunoassay. ELISA was discovered in 1970. It was discovered in 1970. Okay. Sometimes in Vivas, it is asked very commonly. So it is required, and it is also a topic in master's classes. Hence, it is very important. So let's discuss history of ELISA. It was early developed in 1970s. Okay, 1970s by Peter Furman. And Eva by Peter Furman. 1970 by Peter. And Eva. They were from Sweden. They were Swedish scientists. They were associated with Anton Schurz and Bock von Wieman, who were from Netherlands. These were from Sweden, and two other scientists were also involved Hoover Anton Schurz, Schurs, Schurz, okay, W. And Bock. Sometimes this person Bock is also at some places called as Bock and Bach. No problem. It is B-A-U-K, Bock. Bock won Weimar. They were from Holland or Netherlands. They were from Holland or Netherlands. Okay. It was designed as a safer and more efficient alternative to the RIV. As I mentioned, it was safer and more advanced, and it has now become a key tool for various tests and quantitative analysis. Now here we will discuss types of ELISA. What are the types of ELISA? Actually, mainly there are four types of ELISA. Which is very easy to learn and remember. Direct, indirect. Direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. And competitive ELISA. These are four different types based on their methods, and each type has unique applications and their own advantages. Now we will discuss each and every topic respectively. Okay, so first, and in this video I am going to discuss all these topics in short because I am discussing mainly ELISA and its type. So you can make notes from this video to write down your answer in long questions. In various question papers, especially in written examinations, each and every topic is mentioned to write as a short note. I will make videos on each topic later on. So now we will discuss each topic in the short form. So let's start with direct ELISA. Direct ELISA. What is this direct ELISA? In this, okay, it is why it is known as direct ELISA, because it is directly used and it is used for the high purity antigens. In this process, antigen is coated into the plate. Process में क्या होगा? Antigen is coated onto the plate, already coated, and an enzyme-linked antibody binds directly to it. As I said, it is very easy to remember. Direct ELISA, antigen, there is a plate which look like this. Actually, there will be 8 wells. 1, 2, 3, 4, 5, 6, 7, 8. And 8 by 8. And antigen is directly coated on these wells. An antibody comes to bind it directly. That's why it is known as direct ELISA. And what are the advantages for this? It is very simple and quick, because we don't need any long procedure. Here the antibody is directly attaching to the antigens. That's why it is known as direct ELISA. Why it is used? It is used to detect highly pure antigens, highly pure antigens, such as in vaccine preparation or some medicine preparation. It is very important, and also it is important toxin detection. Okay. Next is indirect ELISA. Next is indirect ELISA. What is indirect ELISA? Here antigen was coated. Here antigen was coated. In this method, till antigen is coated onto the plate, followed by primary antibody, followed by a primary antibody, then secondary enzyme-linked antibody. Okay. Now, what is the difference? Just remember, initially there was a plate coated with antigens, and just to put down the samples, antibodies directly will bind to these plates or antigens. Here, antigen is coated as earlier, but now the case is changed, as a primary antibody is already coated. Now the secondary antibody will bind to this plate or antigen. This is the reason why it is called as indirect ELISA. Here the point is to be noted. Plate is coated with antigen with plus primary antibody, and then secondary antibody is applied. Or sent to bind. What is the advantage for this test or this method? It has higher sensitivity. It contains higher sensitivity because it has more signal amplification, due to which the ELISA reader reads precisely, and it is used in most commonly HIV testing, autoimmune disease and antibody research. Okay, it is used in HIV detection, most commonly used, then autoimmune disease, and mostly in research, which are being done on antibodies. Next is sandwich ELISA. I hope you are understanding all these points. If there is anything left to be understood, just drop a comment. I will clear it out and hope you will find your answer. Next is sandwich ELISA. As it is clear from its name, sandwich, two breads and a patty between them. Similarly, two antibodies, one captures the antigen and another detects it. Here, this is antigen and these are एंटीबॉडी घेर के मारते हैं ऐसे वाला, एक पकड़ लेगा तुम्हारे, ठीक है। तो एक इनमें से एंजाइम लिंक्ड होता है, एक इसमें से एंजाइम लिंक्ड होता है ताकि वो उसको पहचान सके। तो इसको सैंडविच कहते हैं इसीलिए, क्योंकि ये दो एंटीबॉडीज के बीच में एंटीजन फंस जाता है। तो यहां पर क्या हो गया कि एक एंटीजन इसको होल्ड करता है और एक एंटीजन इसको डिटेक्ट करता है, ठीक है। तो इसीलिए इसको कहते हैं सैंडविच ELISA, एज आई मेंशन। Why it is important? Because it is highly sensitive and highly specific. This is the benefit, and it is most commonly used in hormone measurement. It is most commonly used in hormone measurement. Okay. Also used in cytokine detection and biomarker analysis. So it is important. Now the next and last is competitive ELISA. Next and last is competitive ELISA. ठीक है. अब यहाँ होता है क्या है, कंपटीशन है. ठीक, कैसे? तो एंटीजन जो है वो कंपीट करता है लेबल्ड रेफरेंस एंटीजन से एंटीबॉडी से बाइंड करने के लिए. यानी एक एंटीजन होगा, उससे दूसरे एंटीबॉडी से बाइंड करना, मगर बीच में एक और एंटीजन रहेगा. यानी दो एंटीजन एक दूसरे से रेस लगाते हैं. तो इससे क्या होता है कि एक खास तरह का एक इनवर्स सिग्नल प्रोड्यूस करता है, क्योंकि वो किसी से रेस लगा रहा है दूसरे एंटीजन से. वहां एंटीबॉडी यहाँ पे एक ही है, सिंगल, बट यहाँ दो एंटीजन है जो कि उस एंटीबॉडी से बाइंड करने के लिए एक दूसरे से कंपीट कर रहे हैं, जिसकी वजह से वो जो एंटीजन है वो एक रिवर्स या इनवर्स सिग्नल प्रोड्यूस करता है, जिससे कि एम्प्लीफिकेशन बहुत हाई हो जाता है और रीडिंग बहुत स्पेसिफिक हो जाती है. ठीक है. एडवांटेज क्यों है, मैंने वही बताया कि हाईली स्पेसिफिक है और स्मॉलर मॉलिक्यूल्स को भी डिटेक्ट कर सकता है. ठीक है. अब इसका इस्तेमाल कहां करेंगे, ड्रग टेस्टिंग में. दवाओं के और जो ड्रग्स होते हैं उनकी टेस्टिंग में यूज करते हैं. टॉक्सिन स्क्रीनिंग में जो खबर आती है ना कि पेस्टिसाइड मिल गया, ये मिल गया, तो उसमें इसका इस्तेमाल किया जाता है. और हार्मोन लेवल एनालिसिस में भी इसका इस्तेमाल किया जाता है. ठीक है. तो ओवरऑल नाउ टाइम टू कंक्लूड द वीडियो. ELISA is a powerful, sensitive and versatile. Okay, ELISA is a powerful, sensitive and versatile technique to detect antibodies. It is used extensively in medical diagnostics, food safety, research and pharmaceuticals. Its continuous advancement are improving its efficiency and accuracy. As I said, ELISA is still being advanced and improved by various companies, and they are working on this topic, and they are trying to make much better ELISA readers for proper and specific detection. This is all about the ELISA. Now in next videos I will discuss each and every type of ELISA in detail. Here they were in summaries, but future and later on I will make video on each and every type of ELISA. Till then, bye bye. Share this video to your friends and subscribe this channel.